Okay, so the last type of transport that we're going to be talking about is vesicular transport. And so just to remind you, this is a video that we watched earlier in this homework lesson, but when we were talking about membrane flow for the cell portion of the lesson. But notice that is a vesicle. It's pinched off an organelle that has a membrane, and it's moving from one membrane location to another, and vesicles are these little bits that have pinched off that helps this movement to happen. And like we said, this sort of vesicular movement can be used to move things, as it's showing here, from inside the cell, and there it moves to outside the cell. And that type of movement is called exocytosis. And then here it's showing the opposite, moving things into the cell, and that type of movement is called endocytosis. So I think X on exocytosis is for exit, or exiting the cell, and endocytosis, the EN, I think we're moving into the cell for endocytosis. But the key thing is that they involve vesicles. Now, there's three types of endocytosis, three ways that the cell moves things inside using vesicles. One is phagocytosis, and this is where they're bringing large particles inside. So this is actually commonly used by macrophages. This is showing a medical animation. Here's bacteria, here's macrophages, and look at that. I'll actually go back and show it again. Macrophages are some of our immune cells. And so again, bacteria, macrophage, and see how that cell membrane moves in a very large way to engulf the bacteria. Here's actually a real thing. Here's the bacteria, and here's a real immune cell, a real macrophage chasing that bacteria. And you can see it's quite the contest. But eventually, we'll see who wins. And there, that macrophage got it. Okay. So that's phagocytosis. Now, pinocytosis is different because the movements of the membrane are not as large. Okay, so here we're just pinching off little bits of membrane, pinocytosis. So these are very small vesicles that are bringing contents into the cell. And a lot of times the cell is going to use this just to sample its environment, see what's out there. It's not trying to take a really large thing into it. It's just sampling what's out there. And then the third type of endocytosis is called receptor mediated. And as you might guess from the name, it uses a receptor. With phagocytosis and pinocytosis, we're bringing anything that happens to be out there in. But receptor-mediated endocytosis means we're bringing only something very specific. And so here it's showing the plasma membrane. This is outside the cell, this is inside the cell, and these are the receptors. And they're going to lock onto and bind. In this case, we're showing they're binding a low-density lipoprotein particle. This is actually how you bring different lipids into the cell, is through that. Okay, and you don't need to know the details of LDL and then it's cholesterol and all of the structure of this, but I do want to show you how this works. So this may be, this cholesterol particle may be in your blood, it comes to the cell, and it docks. Notice how this fits together like a lock and key. It's very specific. Okay, and so once that happens, now notice endocytosis. That is brought into the cell in a vesicle. Okay, so let's go here for a minute to our notes, and let's go back to this chart here and remind ourselves. Now hopefully you know passive and active from what we talked about on previous pages of the homework, but just in case you're confused at all, passive and active do not require vesicles. It's just a protein stuck in a membrane that lets things through. So none of these require vesicles. The only one that requires vesicles is vesicular transport. So is it moving things from high to low or low to high? Well, with vesicular, that doesn't really apply. It's bringing lots of stuff in and out, and so it's not really concentration based. Passive and active are based on concentration, but not vesicular. Is energy required? Yes, to get those big movements of the membrane, you have to have energy and you have to have proteins. Okay. So that is the section on both cells and cell transport. So I would suggest you go through the notes packet, you go through the homeworks, and contact your instructor with any questions at all.